Awesome. Thanks so much, everyone, for being here, and thanks especially to the symposium organizers. This is a symposium about function and form, but right now we're going to swap those with ecology and morphology. And I'm going to talk about some of my PhD research at UC Berkeley, looking at ecomorphology and resource partitioning in spiny lizards. So when I started my dissertation, I was kind of obsessed with phenotypic evolution and looking at the diversity of life. And I was really curious about what drives phenotypic evolution and what the consequences of this novelty, phenotypic novelty, are. This is obviously a huge question, and I did not answer it in my PhD, but I want to focus on sort of one mechanism that we know can drive phenotypic novelty, and that's resource partitioning. And we have lots of examples of this in nature, right? So anolis lizards, one of our great examples where we have partitioning of the vertical habitat space associated with morphological adaptations such as body size and limb and digit lengths. Darwin's finches, where we know partitioning of dietary resources leads to the evolution of novel beaks morphologies, and stickleback fishes, where we have the repeat evolution of benthic and limnetic forms. So from these examples, we can see that resource partitioning is a really powerful mechanism that can both drive adaptive divergence, and then for the consequences side, we know that this can reduce competitive overlap and have really important implications for species co-occurrence. So if we take any one of these examples, I'll take the lizards, we can say that this relationship between these adaptations that we're seeing and where they're perching in the tree or their general ecology is known as ecomorphology. And if we pick any one of these forms, when we see this being evolved repeatedly, we call those ecomorphs. A lot of our studies of resource partitioning and ecomorphology take place on islands. And these are really convenient systems for this type of work because they're often far from the mainland, they might be isolated, they might have limiting resources, and because of that, we expect forces of selection to be really strong. This is also true on non-island islands. So these are sky islands, these are mountains that are enclaves of habitat surrounded by a desert sea, and we expect the eco-evolutionary dynamics of species living on these sky islands to be very similar to what we would see on oceanic islands. And this is where the first part of our talk will take place, is here in southeastern Arizona in the Madrean Sky Island region. So now I'd like to introduce you to my study system, and I think that this is a really great system for studies of ecology, morphology, and resource partitioning because they occur across a wide variety of landscapes and they reveal ecological and morphological diversity. They co-occur really readily, which makes it convenient to go into the field and study their interactions. And there's quite a lot of them. Some people here study really spectacular radiation, so I know 100 species is not that many, but I'll take it. It's a pretty good size for a PhD. So today I want to focus on two major levels of organization. We're going to start sort of at the community or assemblage level. And to answer this question of what is the relationship between ecology and morphology of co-occurring species, I wanted to start by asking what is this sort of major axis of resource partitioning, what's the ecology part of that function, right? And then get into the morphology. And then we'll zoom out and ask across the whole radiation of Scoloporus lizards, so all 100 or so species, and think about how is that relationship between ecology and morphology across species and across the whole range of the genus. So let's go to the field, the Chiricahua Mountains in the far southeast of Arizona, where we have these 4 species of spiny lizards that co-occur in different combinations of syntypy as we go up the mountain. So it's kind of helpful to remember, I'm going to use these colors to depict these species throughout the talk, and it's helpful to just remember that the larger species are in blue, and the smaller species are in these sort of tans. And over the course of many field seasons, I quantified resource use along these 4 major axes: structural, temporal, dietary, and thermal. We'll start with the structural niche, and what I mean by this is perch height and perch type. So I'm just going to cut to the chase and tell you we found some really dramatic differences in substrate use between these species. So we find that our bigger species in blue tend to perch quite a lot higher, and this is true across all of the different sites that we worked in. And so not only do they perch quite a lot higher, but they like to perch on different types of things. So we can see that our species in dark blue really likes rocks. Our species in teal is kind of more tree focused, and then our species in orange really likes bunch grass, that's BG, and shuttling on the ground between bunch grass clusters. When we looked at activity time, so we did standardized surveys from 6 a.m. to 6 p.m. to try and figure out when these lizards are active. We didn't find that many differences, but we did see that between our 2 larger species in blue, they were active at very different times. And they were actually the only pair of species in this whole system that didn't differ in perch height. So we think that this is actually a function of competition for space probably as well. They like the same perches, so they have to partition the time that they use them. Moving on to the dietary niche, so we use DNA metabarcoding to get sort of a snapshot of dietary composition for all of these species across the active season. And it's helpful to know that in squamate reptiles, we have sort of two major foraging modes. We have wide foragers, and then we have sit and wait or ambush predators. And Scoloporus fall into the second category of sit and wait predators. So let's take a look at the dietary diversity that we recovered. So these are represented by operational taxonomic units grouped by order. And each of the bar lengths represents one diet item that we recovered in the diet for the different species. And so when we look at dietary breadth, this ranges from 0 being specialist to 1 being generalist, and we used a couple of different metrics to look at this. What we can see is that these are more or less dietary generalists. They're eating quite a wide variety of items, right? But when we look at dietary overlap, which also ranges from 0 to 1, we found strikingly little amounts of overlap. And a lot of these lizards are like right next to each other, like one is on a rock and one is on the adjacent tree. And so if we look at the blue and orange pair, Yerevi and Sleveni, they had not a single OTU in common, which is pretty striking. So finally, the thermal niche, we looked at substrate temperature, body temperature, and operative temperature. And today I'm just going to focus on that last metric of operative temperature, which is sort of, you can think of it like thermal equilibrium. So this is the temperature that a non-thermoregulating organism could achieve based on its properties, so its size, shape, absorptivity. And the way that I measured this was by cutting copper pipes to size to match the 4 different lizard species and spray painting them to match the reflectance of the dorsum, and then putting temperature loggers inside, setting them out in the habitat and leaving them alone for this sort of peak activity period, and then using that to figure out what temperature could these lizards achieve in their respective microhabitats. So what I want you to take away from this is that these lizards can achieve really different temperatures. Operative temperature was really different. And again, we place these really close together. So this is just a lot of difference in a very small area. And then we overlaid preferred temperatures for each of the lizard species. That's the red bar. And so not only are they experiencing different temperatures in their microhabitats, but they're achieving high thermal quality in their preferred microhabitats. So for most of these species, there's one sort of glaring exception, but for most of them, they're achieving temperatures really close to their preferred temperature in their respective microhabitat. Okay, so with all of that, we can say, our ecological data from the system tells us that species, these co-occurring species, are primarily partitioning the habitat space. So we have an arboreal species, a saxicolous species, a terrestrial species, and a vegetation specialist. By virtue of partitioning habitat space and being sit and wait predators, they're also partitioning diet items and thermal resources. Okay, great. The nice thing about having all this data in the same system is that we can compare the effect sizes, and we can see that while all of these axes are important, perch height and perch type are sort of the most important. And so to get to the second part of our question, what is that relationship between ecology and morphology? I additionally took morphometric measurements for all of the lizards in these studies, including body size, head dimensions, limb and digit lengths, and scale size, and looked at dorsal color matching. So when we put all of those traits into a principal components analysis, we can see that different species look different, which is always reassuring. We found that body size contributed to a lot of these differences, especially on PC1. And then I looked at body size corrected residuals for the rest of those traits and found a couple of other standout traits. So limb length seemed to be really important, and as well as dorsal scale size. Okay, great. What does that mean in terms of what these lizards are doing? So we found this really strong relationship between perch height and morphology PC1. PC1 was body size loaded negatively, so what this is actually telling us is that bigger species with longer limbs are perching higher. And that's really cool because we know that limb length in these lizards and related lizards is really associated with climbing ability and sprint speeds. So this makes a lot of sense with what we're seeing from the ecological standpoint. I looked at a lot of these individual traits additionally and found pretty similar relationships. I also looked at dorsal color, so I took quantitative color measurements for all of the lizards and plotted them in 3D color space. And what we can see is that there's a lot of variation in brightness between these species, which is pretty cool because a lot of predators use brightness to detect motion. So species have different colors. And then I additionally looked at the color of the perch site for every individual lizard. And what I found was that for these two smaller species, these are species with shorter limbs, remember, so they're poorer climbers and slower overall. They were significantly color matched to their habitat, which suggests to me that they're probably relying more on tactics like crypsis to avoid predation, whereas these better climbers might be able to shuttle to the other side of a tree or lodge into a rock crevice. So from this section, the main takeaway is that species primarily partition habitat, and to me, the really cool part is that they're adapted to do so. They have these associated phenotypic traits that allow them to do that. So now let's zoom out across the genus and ask, what is this relationship between ecology and morphology between species and across the whole range? So for this, I relied a lot on literature and field guides to put these different species into ecological categories. So in addition to the 4 categories that we saw in the field, I added 2 more, a generalist category, which we kind of always need, and then there are a couple of species that are really adapted to sand lifestyles. And then I visited different museums and took a bunch of morphological measurements and put those all together. So these are some of the measurements that I looked at. Okay, that's a lot. Let's look at this on the phylogeny. And so what we can see here is I've colorized these bar lengths at the tips of the tree by ecology, and the relative length represents body size. And then I've pulled out just a couple of examples that I think are, exceptionally nice and pretty, and demonstrate sort of some of the convergence that we're seeing between these different ecomorphs. So when I looked at phylogenetic signal, I found, a moderate to high level of phylogenetic signal in morphology, but ecology was not significant. So that suggests to me that morphology might be a little bit constrained, whereas ecology might be a little bit more labile. And this radiation is really a system of both convergence between some of these ecomorph classes and morphological conservatism. So let's look at this in phylomorphospace, and we'll just walk our way around it. So starting in the bottom left with the smallest body size and the shortest limbs, we have our vegetation specialists. Then we have our sand specialists sort of adjacent to them, our terrestrial ecomorphs clustering pretty well together, our generalists breaking all of the rules and showing lots of variation on both PC1 and PC2, classic. Our saxicalous morphs, which is pretty interesting because we have sort of 2 distinct phenotypes within this grouping, in the stem group and then in the rapid radiation, and then our arboreal ecomorphs. So I wanted to know how strong this relationship was. I used a clustering analysis, a discriminant analysis to try and pull apart ecogroups by morphology and found that it did a pretty good job. I did an ancestral state reconstruction to try and figure out how all of this phenotypic diversity has emerged. And the one thing I just want to highlight here is that we have this transition to arboreality at the base of the rapid radiation that seems to beget lots of other ecological and morphological novelty. And when we look at transition rates based on stochastic character mapping, we can see that arboreality is the source of more transitions to every other ecomorph than any other group. Okay, and then if we revisit our phylomorphospace, we can see that arboreality really opened up quite a lot more morphospace in the larger body size, longer and shorter limbs, which is pretty interesting. All right, so the last part of our last question is, so what does this mean for species interactions and species distributions? So we can map ecomorph richness, which is fine, but we really need to contextualize it with species richness, right? So when we look at species richness, okay, these 2 are obviously really strongly correlated. What we're really interested in is how many ecomorphs do we have in a given area based on our expectation from species richness. So we can do a spatial regression of ecomorph richness on species richness and look at the residuals to answer that question. So what I'm going to show now is in red will be positive residuals, which means we have more ecomorphs than we're expecting. White will be as expected, and blue will be a negative residual, fewer ecomorphs than expected. And what we can see is across the range, we have largely positive residuals. We have more ecomorphs than we expect, and so we think that something like limiting similarity might be restricting the ability of similar ecomorphs to co-occur. Okay, so you'll just have to trust me because I'm running out of time, but I shuffled this, and this is a really strong relationship. And when we look by different ecomorph classes, we can see that there are some differences between co-occurrence patterns, and so we can see that some ecomorph classes might be more restricted in their ability to co-occur with one another. So we found a strong relationship between ecology and morphology across the radiation, and that ecomorphs are spatially overdispersed. And looking at different scales from individuals to populations to the whole genus can tell us a lot more about these relationships. So thank you to all my collaborators and the organizers.
Thank you, Erin. Hi everyone, I'm Gokul. I'm a final year PhD student in Dr. Kavita Ishwaran's lab at the Indian Institute of Science. So yeah, just going ahead. So we know that organisms deal with the economics of resource allocation. So the resources that one aggregates throughout their lifetime is invested in morphological growth, then you have performance coming in, you have dealing with immune responses, and you also invest in reproductive traits, all the while dealing with survival risks, right? And the aim of all these investments is essentially to maximize one's fitness, and therefore when expendable resources are limited, heavy investments in certain traits can obviously trade off in investment in other traits, right? So in that case, why are some traits exaggerated? So you have the major claw and these fiddler crabs to the left, and you have the antlers on the ears to the right. And one of the reasons why traits can be exaggerated is that they could either offer you dominance during competition, or they can gain you better mating benefits, right? And typically traits that show exaggerated investments have a positive allometric scaling. This means that as body size increases, the trait size disproportionately increases much greater rates. And this is assumed to be because larger individuals typically have a better body condition, so therefore there's a lot more resources available, and then they can invest heavily in a trait. The fundamental assumption that exaggerated traits are present is because that the relative increase in the trait can essentially increase your fitness quite drastically, right? But there are obviously costs of exaggeration, right? So you have these Irish elk, which is one of the most famous. So they have these large antlers, and one of the leading hypotheses is that the antlers were so maintaining the metabolic energetic costs of these antlers were so huge that that could have been one of the reasons why they've gone extinct. And because of these exaggerations, you can have possibly higher predation risk, and also negatively affects your longevity as well. So the part of my thesis essentially aims at answering why organisms exaggerate, and here I'll be answering how positive allometry is maintained by looking at lifetime fitness in a short-lived, free-ranging agamid species. So yeah, this is the hero of my study. This is the peninsular rock agama, or the Psammophilus dorsalis. This is a rupicolous endemic species found in India. They have a socially polygynous mating system, and they are sexually dimorphic. Individuals have a lifespan of around 1 year in the wild, and they have a single breeding season that spans from May to the end of October. Demographically, we see that most of the areas, it's heavily female biased sex ratio, but operant sex ratios are clearly male biased. And during the breeding season, you have male territories that can change in the centroid and overlap multiple female territories that are mutually exclusive from each other. And both the males and females show dynamic color changes and aggressive behaviors towards their competitors of the same sex, and also show interaction with mates during the breeding period. Right. So we studied these lizards in Rishi Valley in Andhra Pradesh, where the habitat is kind of you have this scrub, thorny vegetation, grassland matrix with these large, flat granite sheetrocks. And we've been following these lizards from for a population from 2008 onwards, where we've been doing regular tagging, measuring these individuals, and also been collecting tail samples for a source of DNA. And we've been also performing life history census by walking around these sheetrocks every week to fortnight to understand what is the demography. And we also looked at multiple sheetrocks that had of varying sizes and demographic factors. So looking at my exaggerated trait investment, which is positive allometry in traits, the first trait that I focused on is head width in lizards. So head width directly correlates with the bite force that a lizard can inflict. So this trait correlates with the complete dominance, and therefore strongly under sexual selection in males. The next trait is interlimb length, that is a proxy for the abdominal volume, and also correlates with the egg carrying capability for a female. And therefore this trait is directly under strong fecundity selection. And last year in evolution, I have actually shown that positive allometry in head width and interlimb did vary in the response to changing environmental pressures across years and space, where head width, a sexually selected trait, varied with temperature, rainfall, and the demographic total abundance that's seen on the sheetrock, whereas interlimb length, which is associated with fecundity, only changed with changing resource levels. And we know that lifetime fitness during the lifetime is aggregated through various processes. So you have to first compete with conspecifics of the same sex, then you have to show behaviors and attract mates of the opposite sex, and also for females particularly, they have to invest heavily in fecundity as well. And thus, the question that I'm asking is pretty straightforward. How is positive allometry maintained? The hypothesis is that greater investment in head width enhances a male's competitive ability, and for a female, it enhances their egg carrying capability. So therefore, greater investment in head width and interlimb length are supposed to increase an individual's fitness quite a lot. We look at it. We look at getting at fitness by using microsatellites. So we started this work in 2020. So Raja, who's a postdoc who did plant taxonomy, and I was a protein biochemist at that time. We started off this in 2020, and we started extracting tails from samples in 2015. We optimized 6 tetra-repeated microsatellite loci, and for our preliminary analysis, we used 45 adults who were 19 males and 26 females from 2015 mating season, and then subsequent generation of juveniles and adults were our candidate offspring. We use Genius Prime to get at the allele diversity, and we use Service for parentage assignment using maximum likelihood. Right. To get at investment under positive allometry, we took in morphometric measurements of adults, that is males and females, during the breeding season. And from there, we essentially performed a log trait to log body size regression to get at relative head width and interlimb length for males and females for a given year. We also know that lizards grow throughout the lifetime, so we need to have a body size that's normalized to a particular day in order to compare individuals. So what we did is we took in data of repeated tagging and measurements of individuals from our study and used it to create a growth curve equation. And from then, we estimated the body size for the peak mating season that's on July 31st for each year. And to get at our final how lifetime fitness benefits for males and females are influenced by the morphological predictors, we have a response variable as number of offspring that is assigned to an individual. And morphometric predictors were body size and relative trait size, that is the head width and interlimb length, which are both used as direct and interaction as well. So first, when we looked at the reproductive fitness data, we have the number of candidate parents. That's to the left, you have number of candidate fathers. To the right, you have number of candidate mothers. And on the x-axis, you have the reproductive fitness, that is the number of offspring assigned. We see that first, the 9 out of 19 males sired offspring, and 18 out of 26 females were assigned offspring as well. Clearly, from the graph, you can see that the variation in fitness is quite a lot for males compared to females. And when we measured the skew in mating fitness, that is the variance over the mean square, we found that it was around 4.8 for males. That indicates a very, very high intensity of sexual selection acting on mates in this population. Right. Now, when we went ahead and looked at how our morphological predictors affected individuals' lifetime fitness, we see that body size actually did not determine reproductive fitness for males. And how to read this graph is you have the fitness on the y-axis, that's the number of offspring. The red dots give you the absolute number of offspring that's assigned from the data. And I've converted my continuous variable, that is body size, in categories as large or small, just for ease of visualization. So we clearly see that there is no difference in reproductive fitness for larger and smaller males. We also did bootstrap just to validate the absence of an effect. Right. When we looked at relative head width investment, we saw that for males who had a larger head width, they did have a greater reproductive fitness. And here what we saw that when relative head width increased by 1 standard deviation, so this is about 2 millimeters for a lizard, what we saw that a male's reproductive fitness increased by about 73%. We also had a strong interaction. We also had an interaction effect where the larger the male is and the larger their head width is, their reproductive fitness now increased by about 150%. But when we did our bootstrap, we found that the 95% confidence interval did not show us a very strong positive effect. So we need to have more data to really be confident of this result. Next, we have body size for females, and we saw that indeed it's the same pattern where larger and smaller females did not have a strong difference in the reproductive fitness. Again, bootstrap also supported the absence of such a strong difference as well. Now, in the case of interlimb length, our results were quite interesting because we saw that the greater the interlimb length or the greater their egg carrying capability had a negative effect on reproductive fitness for a female. That is, for a female whose relative interlimb length for the body size is greater by about 6 millimeters, their fitness dipped by about 35%. And when we also added in the interaction effect, that is when a female is larger and has a relatively longer interlimb length, their fitness now dipped to about 60%. We also ran bootstraps, and here we see that there's a strong, significant negative effect of female interlimb length on their reproductive fitness. So from this study, we infer that weapon exaggeration may have a role in enhancing your mating opportunity and number of offspring. And I feel that a lot more to do with, I think there's a lot to do with how this trait influences behavior and performance during competition, and also in how individuals acquire territories. In the case of females, we saw that greater egg carrying capability actually had a negative effect on their reproductive fitness. One possibility could be that having a longer interlimb length actually negatively affects their climbing ability. So this could potentially have, having a longer interlimb length could potentially have greater survival risks for females. And last, we see that body size had very negligible effect here. We know that like reproductive fitness is a lot constrained on your space that you're living in, right? Like you might not encounter mates and competitors, or you might not even encounter mates in the territory that you're living in. And when we looked at the spatial dynamics, so this is just like a preliminary result. So we have two sheetrocks that are like about similar area. So GDB to the left is a sheetrock of about 1 hectare, and most of these territories have similar male to female abundances. But another sheetrock that's tentrock, which is about like 500 meters away, has really separate male and female clusters. So what could happen is that we are thinking of looking at traits now in point of view on how they affect how you acquire territories and how it affects your behavior as such. So the future focus now is to first obviously get more and more data for stronger inferences. Next is to compare the fitness and selection gradient across a 5-year gap by using data from 2021 and 2022. So this is particularly interesting because we had a strong dip in the mean summer temperature by about 1 degree Celsius, and the rainfall during the summer increased about 1.5 times. So we wanted to see what happens when there's a strong dip in temperature and a greater increase in rainfall during the year. Next, we want to look at how trait exaggeration is heritable by looking at parent-offspring pairs of the same sex. And last, to understand how traits influence behavior, then how it affects what territories you acquire, etc. I want to use the data that I have on focal individual sampling and simulated intrasexual competition by using models to understand essentially why organisms exaggerate investment. So I would like to thank Kavita, who's my PI for support, Rishi Valley NCF for field logistics, Raja, who was a postdoc who worked with me in the earlier stages, Tenzin, who was in project assistant, my interns, my university, and thank you all.
So today, I'm going to talk about the scaly curse of dimensionality, right? Scaly because I study scale-eating pupfish. And curse, I mean, this is kind of Wikipedia's definition, right? But many of the things that we study, genotypes, phenotypes, they quickly become out of control as the dimensionality goes so high that even if we spend our whole lifetimes collecting data, we can't answer these questions in high-dimensional spaces. We can't perceive them. Our perspectives are distorted by this sort of three-dimensional view that we have as a species. So for example, take Wright's classic genotypic fitness network shown here. This is just 5 SNPs and showing all pathways through haplotype space to get across that space, right? So that's 3 to the 5 possible haplotypes, 243 possible paths through this 5 genotype space. But even if we just consider 1,000 SNPs, that's 3 to 1,000 possible haplotypes. That's 10 to the 477, right? That's more stars in the observable universe, more sands than in all the oceans. So we can't actually measure the fitness of all these possible haplotypes in even the simplest populations that we study. Then there's this issue of many-to-one mapping, right? Classically, Steve Arnold's many-to-one mapping of genotype to morphology to performance to fitness, we can think about how these performance and fitness landscapes are changing through time and across a phylogeny. But this one part is the part that I have issue with, right? Of course, we can almost never measure that one lifetime reproductive success fitness estimate we want. We have fitness proxies. So in reality, it's more like many-to-many mapping, right? And I want you to be overwhelmed by this slide, and that's kind of the the sort of thesis of my talk today is just kind of how overwhelming it is when we try to connect these different biological levels of organization. Because there's so many different performance tasks, there's so many different fitness proxies and environments that this is sort of closer to the reality that we're trying to study. I study adaptive radiation, the rapid evolution of new species, and this radiation I'll talk to you about today kind of sits on these two axes here where there's a lot of morphological disparity, there's a lot of ecological divergence, and species richness, it's 3 to 4 species. So they're not super diverse, but they're the beginnings of an adaptive radiation. I also tend to focus on rare origins of novelty and adaptive radiation. So systems where we only see adaptive radiation in one place with lots of other control environments where it hasn't happened. And so first I'll introduce to you today the natural history of Caribbean pupfishes. Then I'll talk about our various attempts to now connect genotype to phenotype, phenotype to fitness landscape, genotype to genotypic fitness network, and finally phenotype to performance landscape. And then how do we combine all these things? I don't know. But this will just mostly be a lot of caveats about these kinds of approaches. So yes, pupfish are named after puppies because they like to play like puppies. They are most famous for being isolated in Devil's Hole, which is in the center of Death Valley. It's the hottest desert in the world. I encourage you to go to my grad student David Tian's talk on Tuesday. It's right in this room as, let's see, 516B. But he'll be talking about our project on conservation genomics of the Devil's Hole pupfish, including this mysterious potential candidate gene called deathbox that might be killing these things, even in managed populations. But pupfish are found all over the Caribbean, from Massachusetts to Venezuela, throughout these Caribbean islands, which they range here in blue. And throughout this range, there's only 2 places where you get closely related sympatric species occurring in the same lakes. And the focus of my talk today will be San Salvador Island in the Bahamas, where these pupfish live in these interior lakes that are about 10,000 years old based on changes in sea levels. So these are the 3 species. They fit in the palm of your hand. All 3 of these are collected from the same benthic habitats, the same net trawls. So this is true sympatry. And they're found there's only about 2 species here, 1 species here. All 3 are in this little sliver of a lake here. So it doesn't even relate to lake area. There's just kind of this hodgepodge collection of sometimes you get lakes that can support all 3. In the wild, one of these species is a specialized scale eater here. So this is filmed at 500 frames per second. 50% of their diet are the scales of other generalist pupfishes. The generalists are eating this algae, detritus. The scale eaters are also eating that, but supplementing that with scales. So here it is in the lab. This is filmed at 1,000 frames per second. And you can see that spray of scales off the side of this generalist. I should say, this guy was euthanized first, so freshly euthanized. Here it is in the lab at Berkeley, freshly euthanized zebrafish. And you can see they're basically scraping off the mucus off the side of that prey. And they're doing this all day long in San Salvador Island. So I named this species desquamator, which means one who removes scales from a fish. Now there's other scale eaters out there, but this niche is exceedingly rare among fishes, which I don't think means it's irrelevant. I think it means it's fascinating how across the deep sea, across Amazon, southeastern Asian streams, we see this niche evolving repeatedly, and we still don't understand why. Why isn't our scale eater in every single fish community on Earth? I don't know. And I mentioned it's not just this one weird specialist, there's a second weird specialist. So this is the molluscivore pupfish. It actually has, this is not its upper jaw, this is a nasal protrusion that sticks out above the upper jaw. It's actually a skeletal protrusion here of this blue bone, the maxilla, that exceeds that upper jaw here in red, the premaxilla. Now there's other fishes out there with nasal protrusions with a variety of functions, but this is the only one within Atheriniforms, which is over 2,000 species with nasal protrusions. Now what we found in the lab, and we think in the wild as well, it actually pulls snails right out of their shells. So this is an oral sheller that eats more mollusks than the other pupfish out on those lakes. And you can see here, but then we tried to see if the nose is involved in that, and it doesn't appear to play a role. So this is still sort of a mystery appendage. And as you might expect, we've done some morphological diversification rates in this system, and relative to neighboring islands, there's pupfish on all these islands in the Caribbean, but just generalists on the neighboring islands. So some of these craniofacial traits like lower jaw length, upper jaw length are evolving over 1,000 times faster than neighboring islands, driven by this exceeding trophic specialization. So, genotype to phenotype. right? This is mostly originally driven by my grad students, Emily Richards and Joe McGurr, my first two grad students when I was at UNC. And first of all, despite these extreme morphological differences, out of about 10 million segregating SNPs in a genome size of 1 billion base pairs, Joe found there's only 157 fixed SNPs between that molluscivore and scale eater that I just showed you, which is really great for an evolutionary geneticist, right? And most of these are probably even false positives, so we think we have a very narrow range of candidate, potentially causal SNPs to sort through. We also looked at structural elements. We only see 87 fixed deletions in the scale eater. And then, to be a little bit more relaxed, we're not saying it's only fixed deletions that are causing these differences. There's also allele frequency shifts. So Emily looked at sequencing across the Caribbean. We looked for regions where there's a SNP that's highly differentiated that shows evidence of positive selection. So in other words, it's in a hard selective sweep. And with that, we could narrow it down to a few thousand SNPs in each of these specialists that show evidence of selection and are highly differentiated. But again, what's wild here is that most of the genetic variation that's contributing to being a scale eater is found on other islands. So we're seeing it all over the Caribbean. So all this part of the pie that's in gray here is genetic variation that we see in North Carolina. We see it in the Dominican Republic. We see it in neighboring islands in the Bahamas, right? And there's only this tiny little slice in red that represents de novo mutations that we've only observed in the scale eater so far, and no de novo mutations that we've ever observed that are only in the molluscivore. So essentially, we're taking pre-existing genetic variation to build these novel trophic specialties. So next, connecting genotype to phenotype, right? We've now done transcriptomics. We've done genome-wide association mapping for some of these traits. We've done sort of F1 allele-specific expression to look for cis and trans. We've done a lot of sort of different approaches. And narrowing this down, what started to fall out was one, my approach now is a very rule of thumb kind of thing. If I see the same gene popping up in multiple dissertations, that's what we go after. And so I'm going to try not to go into gene land, but this was our candidate that kept popping up in one of my QTL, where it explains 15% of the variation in jaw length. There's a QTL here. Of course, there's lots of genes in this region, but one of them was GALR2. And then in a separate study of the genome-wide association mapping, we see GALR2. So it's differentially expressed. It's evidence of positive selection. And it only has 2 upstream highly differentiated SNPs that are in the regulatory region of this gene. Just 2, right? Again, which is the benefits of this system that is so genetically similar, right? And so we're not sure if one or both of these might be functional. We're trying to look at that now. But here's where my fantastic postdoc, Fania Palominos, comes in. So she's been in my lab for the past 2.5 years. But first, she confirmed that this gene is involved in craniofacial development, which had never been shown in a vertebrate before. Before this, it's a receptor for the hormone galanin. It's known to be involved in appetite. And we see this sort of in green here is GALR2 expression in the lower jaw. So it really looks like it's expressed in chondrocytes. And we have pretty solid evidence now that this is craniofacial gene. But then for the phenotype, we could inhibit the GALR2 pathway because there's a lot known about this pathway. There just happens to be a chemical here, M871, that can inhibit only GALR2. And only in the scale eater background, we found that if we inhibit this pathway, we get much smaller jaws, right, relative to the uninhibited here. But then you might be saying, okay, you just told me about hundreds of loci and this tiny little, if you squint, you can kind of see a difference here, right? And so for all evolutionary geneticists, right, the elephant in the room is what you should be thinking is effect size, right? And so effect size here is potentially as high as 15%, right, which is that QTL. But that's certainly an upper bound. And my worry, of course, is that it's less than 1%, right? And so we really don't know where it is with effect size yet. So that's where the other part of this gene story is that it's a hormone that's involved in hunger, so think scale eating. It's involved in nervous system development. And now we've shown it's involved in craniofacial development, right? So this is one-to-many mapping, pleiotropy. But lots of what we're thinking about here, we can't really think about these genes having one function. Of course, they have many functions. And then we have lots of other craniofacial candidate genes to go after now in this system. how many of these are affecting just all of the functions that we're interested in, and how can we even isolate these things? So kind of when I think about connecting micro to macro, I'm thinking about for each of these black or highlighted sort of adaptive candidate alleles, we can estimate their age in time. We can estimate whether they're de novo variation that's only found in the scale eater, whether they're found everywhere in the Caribbean here, or whether there's introgression from specific sources. But then we can functionally look at their effects on phenotype, on performance, on fitness. But then there's many-to-many mapping. And so how are all of these things interrelated to each other, right? And so that's the 40-year plan here for the rest of my career, right? We do have CRISPR working now in this system, so we made a pale pupfish here. But the hope is to start going after these candidates one by one. So that's the genotype to phenotype story for just one candidate. What about phenotype to fitness landscape? Now, I'll go over this section fast because I've presented this at Evolution many times before. I think some of you have heard it. But essentially, if 98% of the genetic variation is the same everywhere, then maybe natural selection is the reason that scale eating is so rare, right? And so to do this, as a grad student, what we really want to do is measure hybrids, not just the wild phenotypes. So I raised all these F1 and F2 outbred hybrid crosses in the lab from 2 different lakes, over 2,000 F2 hybrids with a diversity of morphologies, right? Basically put implanted coded wire tags, took photographs, every single fish, built these field enclosures back in the original lakes of low density, high density, 2 different lakes, right? Inside looks similar to outside, take pictures of all the purebred parentals in the lab as well, so that I can kind of get a morphospace to sort of anchor what those hybrids are looking like, right? So this is a discriminate function space just showing you the separation among these 3 different sort of wild-caught and lab-raised individuals of the parents. Then within this space, we can plot the deaths of those F2 hybrids. This is after 3 months in that first high-density enclosure, and also the survivors after 3 months. And already you can kind of see some real dramatic differences in survival across this morphospace. So this is that fitness landscape, connecting phenotype to survival in the wild for the first high-density enclosure, right? And what's wild to me here still, right, is that there's a small fitness valley separating the generalist from the molluscivore phenotype. So these are hybrids resembling the molluscivore phenotype, essentially with that nasal protrusion, have much higher survival. So there's essentially 2 different ways to survive in that tiny little enclosure, whereas the scale eaters just do miserably bad, right? And you should never, ever repeat a good experiment, right? This is my advisor at the, so for my postdoc, and Sam Price is here too, I think. But she was like, just do it, Chris. So I repeated this experiment for my postdoc. I tried to do it better, right? 4,000 hybrids, they're fin clipped, frequency manipulations. This is larger enclosures. Long story short, there was a hurricane. I'm not going to go into more. We did recover data, but it was a much kind of less of a strong signal here. This is basically using generalized additive modeling to estimate thin plate splines across all the enclosures. This is kind of an averaged, what does that landscape look like? But if we compare to sort of the average estimate and some of the original fitness landscape measures, what was still surprising to me is that these peaks are in the same place in morphospace. So there is some stability there. So at this point, you might be thinking, okay, great, that's encouraging, right? Like there's one-to-one mapping here almost of this fitness landscape. But I want to also emphasize, in case we get too sort of proud of ourselves, right? There's no single landscape. This is basically showing, this was a slice of 2 dimensions out of just the 16-dimensional space that I measured from those traits, right? Whereas, of course, with phenotypes, maybe that's a million-dimensional, a billion-dimensional space, right? But we can take a 16-dimensional space from the 16 traits that I measured and just look at what these landscapes look like for a whole bunch of different 2-way interactions. So these are essentially all the nonlinear interactions, and you can see that there are single peaks. There's one that has 3 peaks. Some of them have 2 peaks like we observed. So even across traits within this radiation, there's different fitness landscapes depending on how you slice up that volume. And now it gets worse. So we also wanted to do what Wright proposed, the genotype to fitness network. right? And so this was work by my postdoc, Austin Patton, who's now works at Arcadia. He left academia because of this, essentially like the curse of dimensionality. So he was, through the pandemic, Austin was working in a small cabin in the Oregon woods on this project, and we were all virtual. He was a ghost doc, right? But we sequenced those hybrids from the second experiment and some of the survivors from the first. So it's only 140 hybrids. But for them, we have their growth rate in the wild, their survival in the wild, so some of them just died, right? We also have their phenotypes, and we have their genomes to about 10x coverage. And this is just showing you that here's the wild types. That's good structure there. There's varying degrees of ancestry across these hybrids that we sequenced, which is good. And this is replicated across 2 different lake hybrid populations. And surprisingly, we were actually able to find, genome-wide association mapping for fitness. We were able to find peaks here associated with fitness and look at what genes are in those peaks. None of these genes are related to craniofacial development, to my chagrin, right? But what was surprising here is when we account for these outlier SNPs associated with fitness, when we estimate the fitness landscape, it actually improves our resolution of this phenotype fitness map. So even with just 140 individuals measured, we went from a flat surface with no genotypic information to this curved surface that, again, is sort of what we're seeing with the much larger field experiment data when we just have 150 hybrids and we're controlling for their genotypes. So it does seem to help remove this background noise of other sources of fitness in the experiments, right? But what we really wanted to do, right, was reconstruct genotypic pathways first from generalist to scale leader. And so again, we sort of looked at this set of about 1,000 SNPs that look like they're under selection, that are highly differentiated between these species. And then from there, we can say, okay, how many mutations separate a generalist from a scale leader in our observed data set, right? And again, that's this many possible haplotypes in that pathway. So we had to basically draw down, and out of 1,000 plus SNPs, we would do 10 at a time. And even with just 10 SNPs, it's taking days and days to estimate this network, where essentially each of the gray dots is a hybrid observed haplotype for those 10 SNPs, right? Each of the blue dots is a molluscivore observed in the wild versus this red are scale leaders observed in the wild. I mean, the kind of good comforting news is, as we expected, and based on phylogenetic data as well, that, scale leaders are more isolated on this sort of genotypic network from the generalists, from the hybrids, right? But here's what's really fun is we also have fitness data for those hybrids. So we can reconstruct the fitness pathway through that genotype space, right? And so we can think about this as what is an accessible fitness path, right? In other words, how do we move from this sort of ancestral state of the generalist, which is what the Caribbeans look like, the pupfish look like all over the Caribbean. How do we move from this sort of tan haplotype here through genotype space, one mutation at a time, to get to a scale leader, to get to a molluscivore? What does the fitness look like along that pathway? And I've just shown you in phenotype space, there's valleys here that are isolating these individuals. But of course, in higher dimensional phenotypes, there could be fitness ridges. In genotypic fitness space, there could be ridges where you can move from a generalist to a scale leader and go uphill the entire way, right? That's the problem of evolution, as Fisher put it, right? So here, we're talking about an accessible fitness path is a path through this mutation space where you increase your fitness at each step. So we looked for those in the data. And so that would look like this. We're moving from a generalist through all these observed, these are actually observed sequenced hybrids from our field experiment, not theoretical haplotypes, right? We can move through all these hybrid haplotypes to get to the scale leader genotype at the other side, right? And by doing this, we estimated that about 2% of genotypic pathways between generalist and scale leader are accessible to evolution. In other words, they're either a ridge or they're increasing at every single step. So there are apparently these sort of hidden to our phenotypic measurements, right? These ways to move through that hybrid space without reducing your fitness at all. And it's a little bit higher, you can see here for the molluscivore, it's about almost 4% for the molluscivores. Now the catch of this is, I don't know what this means. Is this number high? Is this low? I've been asking some of you. We didn't know what to expect going into this. There's some work by Patrick Nossel that looks at sort of the color polymorphism locus in walking sticks, but we have no other animals to compare this to, right? And so to me, this is pretty high. But I'd love to sort of see more studies like this so we really know. And it does say that this massive valley that we always observe for the scale-eating phenotype, that there is actually a genotypic pathway to that phenotype. Now, the really weird thing is we can also compare the source of genetic variation. And if we just look at standing genetic variation, there are fewer accessible pathways to those specialists than if we look at de novo and introgress variation. So it does look like this, essentially that de novo, it's flattening the landscape to get to these specialists, right? That's kind of the only way, flattening or maybe making the paths shorter, essentially making it easier to access these dystrophic specializations. So just another way that hybridization facilitates adaptive radiation, right? So finally, phenotype versus performance landscape. So this is recent work in my lab. So originally by my grad student, Michelle St. John, she found that we can measure scale biting by just measuring the areas removed from these delicious Repashy gels that are commercial fish food diets. We've also looked at actually biting on those euthanized fish. It's really hard to actually tell if the scales are removed. So this is sort of our best proxy. The kinematics of those two different types of strikes look pretty similar. And we sort of had a hint that there might be a fitness peak, or sort of performance peak here for biting performance. But I wanted to follow up on this. So I had a team of scale biting undergrads just running videos. I particularly want to highlight Anson Tan here, who he did this project for my fish class, where I teach a research-based ichthyology class. Then he followed up over the summer, and he proved to me that automated tracking would work for these videos. First with deep lab cut, then we switched to sleep. So this is the AI placing the landmarks after me training about 800 videos, 800 just images. So it's really quick. I mean, it takes about an afternoon to landmark, and then we can hit enter and score all 200 plus videos overnight on my laptop. And so these yellow landmarks are placed by the AI. And so it's even getting like lower jaw joint and things like this, right? Let's see this one more time. So this is a scale eater. These videos are filmed at 1,100 frames per second. So these strikes are faster than you can blink. And sometimes you can even see the little bit of gel that they pull off. Now then we basically take that gel out and we immediately, with calipers under a stereo microscope, we measure how much was removed just from width, length, and depth, which is difficult. But essentially, we can take these kinematic variables now from those strikes, which are sort of the classic variables that you may have heard about from Catherine Korn's awesome talk this morning, right? But things like time to peak gape and ram speed and peak gape, right? And peak jaw protrusion, and associate these with the bite dimensions that are removed from these gels, which these are some of the larger gels, right? These are larger bites. These are tiny little pupfish. But we have over 100 strikes for this project now from 3 species plus the hybrids that we had in the lab. And first of all, I just want to show you, this is a discriminant morphospace for the species that are in different colors. So unlike the earlier one from the field experiment, we cannot discriminate the species by their strikes. Their strikes are all over the place, right? So one individual phenotype can essentially nearly generate across the entire performance space. And the same is true for the type of strikes. We can't even discriminate if a strike was a miss or a hit. Sometimes they just touch the gel and just kind of bounce off. The kinematics is not, it's about 50% accuracy to distinguish those kinds of strikes. Really poor, right? But then with generalized additive modeling again, where we can actually fit splines to this data and evaluate whether the splines are a better fit than just sort of fixed effects terms, what jumped out of this was that there's a thin plate spline model that's the best fitting model that has an interaction between the peak gape and peak jaw protrusion. And most of the other terms are either not in the model or they're linear. But what's wild here, 2 performance peaks for scale biting, even in this really sort of simplified laboratory model system, right? And you can see this is kind of robust across. This is the bite length. This is the total gel volume removed on this side, and then whether we include the missed strikes or not. Like this, the 2 peaks are pretty robust to different combinations of variables. And what we're seeing then, and it's kind of a little bit hard to see here, but there's essentially red dots of the scale eater are all over the surface. So scale eaters are performing strikes that are within the range of both of these peaks. the hybrids that have scale leader ancestry, and we actually had both kinds of hybrids, only those hybrids are also in this second peak where it's larger jaw protrusion, larger peak gapes, right? Oh yeah, still have some time. And then, so essentially what we're finding is that these generalist molluscivore and their hybrids are only able to perform strikes within range of this lower sort of small peak protrusion peak. Whereas the scale leaders and their hybrids can do whatever they want. But they're the only ones that are able to occupy this higher performance peak. And this, I mean, this is the actual empirical estimate from that thin plate spline surface. This is not a simulation, right? So of course, to sort of conclude this talk, and what I have been wondering for most of my career, right, is how do we overlay these surfaces relative to each other, right? You heard 2 peaks. Here's 2 peaks on performance space. Here's the 2 peaks from survival space in the wild. Yes, there is 1 peak for generalists in both of these spaces, but at the same time, that second peak is the scale leaders for performance, and in the wild, that second peak is molluscivores, right? And even if we tried to overlay, you know, 1 individual here can actually perform most of the range of this space. So these are not the same axes at all. It's not yet clear to me exactly how we map these different spaces onto each other, right? But certainly as Joseph Wada and Catherine Korn are doing, like, we can use these to constrain macroevolutionary diversification. But really across these levels of biological organization, I'd love to talk more about how do we actually combine all this data into one estimate. And just to give you an example, right, here's the range of the performance space occupied by generalists. Here's the range occupied by scale leaders. So pretty dramatic difference in that kinematic flexibility. So wrapping up, I've kind of shown you that, you know, this is the goal, right? Connect genotype to genetic pathway with this GALR2 inhibition. We've looked at development with in situ hybridization. We've looked at phenotypes and their performance through the gel biting. We've looked at fitness landscape growth and survival. We also have looked at reproductive isolation in this system and rate of diversification. So thank you all for coming.
And our next talk here is going to be pre-recorded. And I will start it.
Hi, my name is Mauricio
Wait. Oh, now. Okay.
Gonzalez Forero, and the central question driving my research is why did the human brain evolve? Now, a major pattern in human brain evolution is the massive expansion in the size of the brain over the last here in this plot. So here we have that chimpanzees now have a brain size that's about 2 times larger than expected for their body size. And a few million years ago, Australopithecines had a brain size that was just a little bit larger. But over time, in this lineage, the brain massively expanded so that humans have now a brain size that's over 5 times larger than expected for our body size. Now, this massive brain expansion is thought to actually have led to large increases in human cognitive ability, but there is a long-standing question into why did this massive brain expansion happen, and why didn't it happen in other lineages to a similar extent. Now, there is a lot of hypotheses to try to explain why large brains evolved, and they can be sort of classified in the 3 categories shown here. Those sort of emphasizing that ecological challenges promote brain expansion, social interactions promote brain expansion, or that cultural interactions, or sort of, for example, having to learn from others and accumulating knowledge in the population promote brain expansion. Now, these hypotheses are very actively tested, and this is the standard approach to test these hypotheses is by using correlational analysis, such as shown in these figures, where some variables are picked as describing the hypothesis, and whether that variable is correlated with the brain size, well, then that is interpreted as supporting the hypothesis. But that approach is problematic because it's a correlation. We don't know the direction of causality. And on top of that, we actually have that, well, if we're actually interested in understanding the evolution of the human brain size, well, humans are essentially one data point among all these species, and that means you cannot actually do these correlational analysis to address the problem. So basically, addressing why the human brain size evolved has been a challenging problem because we lack research tools to infer the causes of actually what's effectively an evolutionarily unique event, because we're looking at the single lineage and the process that happened over that lineage. And so we are at a loss. We don't have tools to actually address that problem very effectively. So I've been putting together mathematical tools to try to address that. And I suggested a promising way forward is what's sometimes called simulation-based inference. And this is an idea that's been actively used across science. And what you do is basically you just take empirical data that you feed into mathematical or computational models that can replicate the event of interest, which here is the human brain expansion. And then you use statistics to see which model best explains the observed data. This approach has been very successful across science. However, it's been notably underexploited to understand why the human brain size evolved. So I've been putting together mathematical tools to fill this gap. And the first step has been to formulate a mathematical model that can replicate human brain evolution, human brain size evolution. And I call that the brain model. I'm not going to go into the details of the model, but basically what the model does is, well, it gives predictions, quantitative predictions for the evolution of body sizes and brain sizes over ontogeny. So the lines, the blue line here is the predicted body size over the life of the individual, and the pink line is the predicted brain size over the life of the individual. And the dots are the observed values for an average human female. So for these parameter combinations used here in these figures, we have that the model actually can recover the evolution of adult brains and body sizes, matching those observed in humans. Now you can actually change parameter values, and these parameter values are actually described by these two elements here. So we have a pie chart, we have a curve here. We don't have to worry about what they mean, but essentially they just describe parameter values that the model has. And using the parameter values indicated here, the model recovers the evolution of adult brains and body sizes of Homo habilis, which have relatively small brain sizes. And if you change the parameter values involved in this pie chart and curve, then you actually can recover the evolution of all these different, of the brains and body sizes of all these different hominin species, all the way to Homo sapiens. And by doing a sensitivity analysis, what you can see is that actually what's causing the brain expansion in the model from the small brains in Homo habilis to the large brains in sapiens and Neanderthals is actually experiencing a challenging ecology and what's called cumulative culture, or basically the ability to learn from others and accumulate knowledge in the population. So what is causing brain expansion here, it's not having to cooperate or compete with other individuals, which has been a leading hypothesis over the last few decades. Now, I wanted to understand more deeply what is causing brain expansion. For example, I wanted to know what is under selection. Is selection actually for brain size that's driving this, and if so, to what extent? However, I couldn't actually address that. And the reason is, well, there's been a long-standing lack of synthesis, mathematically speaking, between development and evolution. And that's a problem that has been in the field for 100 years. Basically, the mathematical tools that we have consider evolution by neglecting development in one way or another. So because the model here actually considers development quite explicitly, I actually didn't have tools to model or to understand evolution very deeply. So what I had to do was to put together a new mathematical theory that integrates development and evolution. I call that Evo-Devo dynamics, and it yields actually quite a lot of insights, filling this gap of long-standing lack of synthesis between development and evolution. And I'm just going to give here the key insight of the theory. So here we have that, well, what a classic evolutionary theory gives, and this is actually from the Landy equation. And what you have here is a fitness landscape, and the fitness landscape for two traits. So we have that if the population is somewhere in the fitness landscape, then natural selection is going to push evolution up the landscape, and evolution stops when the population reaches the peak of the landscape. Now, what development does is that it creates genetic covariation between the traits so that it diverts evolution in some direction, but it actually just, it is just a transient effect. Eventually, the population just converges to the peak of the landscape. So in that sense, development has minor evolutionary effects in this theory because it just has a transient effect by diverting evolution in some direction. You always end up you're reaching some local fitness peak. Now, in this new integration of development and evolution, what this shows is that, well, actually, if you want to understand the evolution of the phenotype as the climbing of a fitness landscape, you have to also follow the evolution of the underlying genotype. And if you do that, then there is a relationship between the two that emerges because of development. That is given by this path. So this relationship, again, given by this path, is sort of the relation between the genotype and the phenotype that emerges over development. And that means that there is genetic covariation exclusively along that path, so that evolution can only happen along that path. And it stops at the peak of this path, not at the peak of the landscape, as it's traditionally assumed. So if there are reasons that, for example, change development intrinsically or extrinsically into some other relationship, for example, here we have a different path, then that changes the peaks of the paths, so that changes the evolutionary outcomes. So in that sense, development has now major evolutionary effects, because it can now change the outcomes of the evolutionary process, even if selection doesn't change, that is, even if the landscape doesn't change. Now, in addition to giving these sort of basic conceptual insights to the evolutionary process, this new integration of development and evolution actually gives us the machinery to understand the evolutionary dynamics of the developmental of the brain model that was actually my aim. And this is what I'm going to show here. So this is the brain size, adult body size for a number of different hominins shown in green squares. And the model is going to start right here. And I'm going to run a video where the model is actually going to show the sort of the evolutionary dynamics of adult brains and body sizes. And it's going to start here. So we see here the brain size starts evolving and it converges to small brain sizes. But then I'm going to change parameter values so that we move to the parameters that actually yield the massive brain sizes of humans. And this is what's happening here. So we see now the evolution of these massive brain expansions converging to species 1, which is Homo sapiens. Now I can zoom in to see what's happening at each of those time steps. So we see here the developmental process that individuals undergo at this ancestral point. So at this ancestral point where everything starts, we see that individuals develop a brain size according to this blue line. These are the predicted development for the individuals for brain size, body size, the ovarian follicles that individuals have, and the skill level that individuals have over their life. And the observed values for brain size in females in humans are actually shown by these black squares. In chimpanzees are shown by these triangles, and in afarenses are shown by these stars. So what we see here is that, well, I'm going to start the model again at this dot, and when we see here that the brain size starts converging to these small brain sizes and it just stays there, and then I'm going to change parameter values that actually lead to the massive brain expansion brain size. And at that same time, we see the evolution, for example, of the growth spurt over adolescence and the long period of childhood that characterize humans. And also we see the massive expansion from about 2 terabytes in adults to about 4 terabytes in adults in terms of their skill level. So what this is showing is, well, the model is actually recovering both the evolutionary dynamics that we observed suggested by the fossil record and the developmental dynamics that we observed in modern humans as shown in these patterns. So the model is doing a pretty good job actually at recovering both things. And now we can use the machinery of the theory to try to understand why this massive brain expansion is happening in the model. And this is what I'm going to show here. So here we have the adult brain size and adult body sizes, and the squares are the observed values in a number of different hominin species. The circles are the predicted values by the model when we have the parameter values indicated by these pie charts right here. So we don't have to worry about what those parameter values are, but what this means is that, well, we can recover the evolution of the brains and body sizes of all these different species by changing parameter values. And now we can actually use the machinery of the theory to prove that actually what these parameter values are doing is only changing genetic covariation. They are not changing selection for anything. So the reason that the massive brains are evolving here from afarensis to sapiens is actually entirely due to change in the genetic variation in brain size. And you can actually go further. You can plot the selection gradient for all the traits in the model and see that there's actually no selection for brain size. There is actually selection exclusively for ovarian follicles, and that's what's actually driving the whole thing. So we see that the reason that the brain is expanding in the model is because the brains are genetically correlated with ovarian follicles. And actually you can go deeper and see that and prove that the genetic variation that drives that brain expansion emerges over development when individuals experience a challenging ecology and cumulative culture. So to conclude, what we have here is that, we have put together mathematical tools to try to infer why the human brain size evolved, and we have a model that recovers brain expansion, and in this model, that expansion happens because of changes in development, that is in the path on the landscape of the fitness landscape, rather than because of changes in selection, that is not because of changes of the landscape. So that means that the effects of experiencing a challenging ecology and cumulative culture that cause brain expansion are actually because of changes in that path that generates genetic variation between ovarian follicles and brain size. So in a sense, what's happening here is that the reason that the brain size expands is because it's essentially a byproduct of selection for ovarian follicles. So that actually matches the notion of Gould and Lewontin of spandrel. Essentially, the brain here is just a byproduct of selection for something else. Now, if you're interested, well, this has just been published, and it's open access, so you can have a look anywhere you are. And with that, I'd like to thank my collaborators, my funding sources, and you for listening. Thank you very much.
Okay, yes, perfect. Thanks a lot. Very happy to present this work with Theo, Melody, and Nicolas. So Melody gathered the data, the mammalian. So it's going to be few mammals data gathered by Melody. Theo and Nicolas, they were in the design, all the processing, and also the interpretation. And so I'm postdoc now at Lausanne, and the question we have is how to detect selections. I'm going to boil it down to several questions, and there's going to be even more questions coming in. And also, as a disclaimer, there's going to be one slide with equations. All of the others, they won't have any equations. But if you want more, there's backup slides with many more. So the question we have is a trait naturally evolving, or is it under selections? And if it's not neutral, is the trait actually changing too fast compared to the expectations, or is it going too slow? And when we're talking about this, or fast or slow, at which scale are we talking about? Is it between species, or within species, between different populations? So this is how fast the trait goes depends on the scale which looking at it. And another question that we have is also how to compute variations and how to normalize it so that it's comparable between actually different scales. So these are the few questions. Here's the data. What does it look like so that we all agree on it? So this is a species tree, and for each species, we might have several traits. In that case, it's not going to be highly multivariate. It's going to be a few traits. And for each of these species, we have several individuals with the trait that is recorded. This might not be all the same individuals for all the traits, but we have variations between and within species. So there's been method developed at different scales to look at whether the trait is under selection or not. Within species, what you typically look at is the differentiation in the trait between the different populations, and you compare the trait compared to the genotype. And for when you look at between species trait variations, what you're looking at usually is how the trait is changing along the phylogeny. And this is what I'm going to focus a bit more here. So how do we model traits along the phylogeny that's changing? So here, like every point here that is changing color, it's a speciation event, and all the two species evolve along different lineages with different trait value. And you have typically, it can be more, but you have typically three models for that. The first one is for a neutral trait, and in that cases, your trait is just shifting randomly up and down. And so this is called a Brownian process. When you want to add selections, what you can start with is actually having an optimal value for the trait. And if you are drifting further away from this optimal value, you get negative selections, like lower fitness. And so this is modeled as a Norsten-Uhlenbeck process. And if you want to have changing selections, in that case what we would call diversifying selections, you would have shifted optimal values that would change along the phylogeny. And you can have different optimals, one, so two or more, and that depends on how you want to model it. So this is typically how it's done, and you usually look at one mean trait value per species. But this has several problems, and the problems here derive from the fact that a OU process can be favored for neutral traits. What does it mean is that you can find evidence for selections while there is none. So that's the first problem. And the second one is actually, it's the opposite, is that you can have a Brownian process, so mimicking the neutral one, that can be favored for a trait under deficing selections if you have changing optima, that you have too many optimas. Meaning that you are in a situation where you can find neutral selection while it's not, and on the opposite, you can find selection while it's not. So this is problematic. And the way we want to do that is that having built an index that will tell you how fast the trait is going, sorry. And so for that, depending on where you are on what you study, so there are the different possibilities here. Not all of them are covered, but that's going to help you build analogy into what we're going to see later. So if you are quantitative genetics across populations, the question that we have is, how could you take a QST-FST test and make that work across different species? If you're more used to PCM methods, the question you have is that for the rate of the Brownian process, what would be your neutral expectation for those sigma? Now, if you're more to our molecular geneticist and you've used DNDS ratio, the question you would have is, how can we actually adapt this strategy of using a DNDS ratio across the phylogeny, but for a trait, instead of protein coding DNA sequences? And now if you are looking more at the contrast polymorphism divergence, and you've used McDonald and Craitman test, you want to do the same thing, but for a trait. And also, you want to have that work across the phylogeny. And so if none of them actually makes sense to you because you haven't studied any of them, you're actually going to learn the analogy with these ones. So it's actually going to be, you're the luckiest one here. You're going to learn even more. So here it's how it's displayed. We're going to go bit after bit, so don't worry. And I promised here no equation so far. So what we can look at first is that between species variations. So this is typically what you want to do is you fit the Brownian process across your tree. Then you have your within species variation. So what you are looking at here is the genetic additive variance, or in other words, is also the phenotypic variance multiplied by irritability. So these are the two things you're looking at. So the first thing you could do, and that this is not novel, it has already been done, is that you can compute the ratio of one of the other. So you're dividing the variations between, and you divide that by the variation within. The problem is here is that if you do that, the ratio you get, it's actually, you don't have a threshold to tell you if it's actually going too fast or too slow. What you could do is actually have a bunch of traits, and this is what typically is done with expression level, where you have 10,000 genes, and you look at the one that actually the highest variance between or variance within. So these genes, typically, it means that their expression level has been changing too fast across the different species compared to your expectation that we see within the species, the variability that you observe in the different inside each species. Okay, so now that we see that, is that what we just need to do is having that normalize so that it looks like a DNDS ratio. And for that, we actually use the sequence divergence on both sides, so the sequences on both sides. So for the between species, what you can use is the sequence divergence, so basically the branch length as measured in substitutions per site. And so what you do is you divide your trait changes along your phylogeny, and you divide that by the branch length. Or another way, you run your Brownian process on a tree that is in unit of branch length as substitutions. So that's the first thing you do here. And on the other hand, what you divide your trait variations at the within species level, you divide that by nucleotide diversity. So basically counting the number of SNPs that you would observe from one heterozygous individuals per site. So that's what you divide. And so no equations here, but we can show that actually when you divide those two and normalize like this, for a neutral trait, and if the sequence are also evolving neutrally, this should be equal to 1. So this is really this rho factor here is like a DNDS. It's the same, works the same way. And so if it's going faster, it means that your trait across species evolving faster than your neutral expectations. If it's on the other side here, you are under stabilizing selections. So that's here for the two bits that are important is really to use the variations between and within, and to use the sequences. So no, like, if there's any questions so far, please don't hesitate. Otherwise, I'm going for the equation slide. Okay, so really quickly, to bore you with this, is that here you divide additive genetic variance by pi, the haplotype diversity, and this discards the mutation rate and the effective population size on both sides of the ratio. And for the variations between, actually what is discarded is the number of generations and also the mutation rate. And so what you see is that both of these ratios are the same, and they are actually the mutational variance of this trait. And so that was it for the equations. Now, how to test that? So we run some simulations. So here is Wright-Fisher simulations where you have diploid individuals and you have a trait that is encoded by several loci, and you make the parents meet, and you have the offspring and selection that's going on, and this is typically what we would look along the phylogeny. And so you want to do that for different scenarios. So the neutral trait, where there's no fitness, whatever your trait is. In case of stabilizing selection, where there's one optimal value all across the phylogeny. And on the right-hand side, a case of diversifying selection, where actually the trait, the optimal trait value is changing all along the phylogeny. And so now when you do that, and you apply the methods that we've found, and here is what is shown across many simulations, the index, the neutrality index that we have. So what we are seeing is that indeed for diversifying selections, we recover an index higher than 1, for stabilizing lower than 1, and for the neutral trait, we are seeing that it's almost close to 1, but it's a bit lower. And what we look actually at the probability of diversifying selections, so here it's in a Bayesian framework, so what you can compute is across your estimations, is your index actually really greater than 1 or not? And so what you recover is that indeed for diversifying and stabilizing selections, you recover what you've simulated. The problem here is for neutral. So that when you simulate under neutrality, what you will recover with your estimation is that a bunch of them will look like stabilizing. What it means here is that you have false positives for detecting stabilizing selections, which is not the case for diversifying. So what it means is that it's a good test for diversifying selections because it doesn't have false positive and also few false negatives, but it's not a good test for stabilizing. So that was a simulation, and just at the mammalian scale, we looked at body size, brain size, and what we are seeing here is that for body size, it looks like almost close to neutral. And for the brain size, which is actually way higher than 1 with very significant p-values, and meaning that this brain size has been varying way too fast across mammals compared to our expectations. So with all of that, what we have is we use the ratio of between and within species. We normalize with nucleotide variations. And again, this is a good test for diversifying selections and not a good one for stabilizing. And so with that, I thank you for your attention, and I'm happy to answer any questions. Yeah, that's perfect.
We have a couple minutes for questions.
I have a clarifying, oh, go ahead. Oh, please. I have a clarifying question. So,
Yes.
first of all, I'd like to say I don't know anything about trait evolution, and you did a fabulous job of teaching it in 10 minutes.
Thanks a lot.
My question is, you need to know species-wide diversification and
Ah, yes, so very good one. Thanks a lot. So just going back to this slide here. So here, the traits should be the same, but the sequences, they should not be the one encoding the trait because they are your neutral expectation. So they should be loci that are evolving as neutral as possible. So for that, and typically what we used in mammals is that they have the whole genome alignment and they have classified which region are evolving as a sequence, too fast, too slow, or neutral. And we use the tree that was built for the neutral one. The problem, and it's important, and it's true one, is that if you don't use those ones and you use another matrix, so actually sequences that are under selections, you're gonna overestimate your trait, your index here. So you need to have those neutral sequences. Thanks.
Thank you.
Thanks. Yes?
Yeah, I wonder, could you see applying this to a gene tree and populations and doing the same kind of test within a lineage?
That would be close to the QST, FST tests that are doing. Does that make?
Yes. Yeah, but I'm wondering if you get the same results on that scale.
Yes, that would be. So that's that really something we want to do. So testing whether what we observe here actually correlates or relates, predicts to what's happening within a species. So typically, if we will do that with brain size, just with these ones, we would not know exactly if it's really predictable. But if we are looking at expression level, let's say across all the genomes, we could see that are the genes evolving the fastest across mammals? Are they still evolving the fastest inside population? Inside the, does that make sense? Yeah, thanks.
All right, let's thank Thibault one more time.
So I've been very generous to put my talk as the last one in my own symposium. So thanks for everyone that is still here. So I'm going to talk about macroevolutionary divergence along allometric lines of least resistance in frog hind-limb traits and its consequence for jumping performance. So we know that most phenotypes that affect fitness are complex. So this means that they are composed of multiple traits that can show a pattern of trait correlations, but might be related to development, to function, or to both. And we can describe this as also being affected by a lot of genetic factors that through development produce these phenotypes. And we know that these magnitude of trait correlations can vary basically because of how this genotype to phenotype map is configured. And trait correlations are very important for microevolutionary predictions because there is a theory that has been developed by Russo-Landi and others in which the shape of this G matrix, which are the patterns of trait correlations, can determine whether the response to selection in red will actually follow the direction of selection. So you can have cases in which the response to selection would actually follow this line of most genetic variants instead of following the direction of selection. So this works pretty well in the microevolutionary framework, but we don't know if this works well for the macroevolutionary side of phenotypic evolution. And this shape of the G matrix, it can be influenced by the adaptive landscape, which is shown here by this red configuration. So in simulations, it has been shown that you can have G matrix instability, meaning that the shape can change a lot in orientation with a shift in the peak. But if you don't have correlational selection, on the other hand, if you have correlational selection in which you're favoring increases and decreases of traits together along evolutionary time, the G matrix can align with the adaptive landscape and be quite stable along evolutionary time. And this is interesting to study macroevolution. So if we're going to complete this genotype to phenotype map that I showed in the beginning, I'm going to add, of course, functional complexes because phenotypic traits do things, right? And what they do is act together, they interact together for perform certain functions that are important for fitness. And so I want to put function, of course, at the center of microevolution. So why? Because if functional relationships are actually determining the shape of the adaptive landscape at the populational level, it can actually determine how populations are evolving, and it can actually make these factors respond to the selection and determine the shape of the G matrix, the shape of trait correlations. So this is a way to put function at the center of shaping how trait correlations would be at the long term. But I also want to connect to macroevolution, right? So why? Because in recent years, we have been seeing a persistent effect of the G matrix on phenotypic divergence. So one of the first people to actually test these kind of things was Dov Schluttler, and he expected, and he saw in his data, that this effect would decay with time, like around 4 million years. But that actually has not been found in several studies, sometimes the mutational matrix, but in several studies that are lots of times using phenotypic matrices as surrogates of G matrices, you can see that this divergence along tens of millions of years is occurring very much aligned with axis' most genetic variants. So this is where you're starting to think that micro and macro can actually be connected, right? I want to test one hypothesis here. There are alternative hypotheses that I don't have time to talk about, which are and this is my favorite, but I'm testing it, so don't worry. Is the evolution along selective lines of least resistance. So what are these? These are major axes of the adaptive landscape. So if you have average trait values here, like tail vertebrate and body vertebrate, and you could have several populations, this is ideal, right? I wish I had this data. If you can have several populations in which you have their phenotypic means, and you can see where they diverge the most, for this hypothesis, it will be along the major axis of these adaptive landscapes in red. So you will see more changes in phenotype along these directions than other directions. And that's because you would expect that adaptive shifts may be favored along these directions because phenotypic changes along these directions cause little or no effect in fitness. So why? Because they're not disrupting, that's the idea. They're not disrupting the operation of functional complexes. So in this case, for example, if you are increasing these traits together, decreasing these traits together, you're not affecting the escapability of snakes. So their fitness are very similar or equal here, but they are diverging. And they are diverging along these selective lines of least resistance. So I wanted to test this. So we could put function at the center of both micro and macroevolution, right? So that's the idea here. So at the microevolutionary level, trait correlations could mirror functional relationships. So you would predict an alignment between axes of maximal variance with function. And at the macroevolutionary scale, you will predict that you will have an alignment of the vectors that are explaining variance in the adaptive landscape with the G matrix. So I'm going to try to test these things. So what data do we need? So it's hard data to get. So fortunately, I have some collaborators that went to French Guiana to collect some kind of these data. So you're thinking they're probably working with snakes, right? No. So they collected data on frog species that occurred in French Guiana, so 7 Boana species, 8 lepidactyl species, in which they can measure jumping performance using force plate. So this is how a curve of the force is going on. So we have the peak, there's the jump. And we could extract data for peak acceleration and velocity. So this is our performance data, right? So I want to show a little video here because it's always fun. If it should work. No. Oh, I'm so sad. Okay. Very high technology. Yeah. So, so this is how they measure it. And so we could have within species morphology and performance. So we have higher numbers of individuals for 2 of the genera in which you measure performance, and then we measured morphology, high limb traits, also muscle constructional area, and I have a simpler data set that I analyze because the patterns are more clearer, so I might show some results on that. And right out the bat, I'm going to say that it was a very similar pattern of trait correlations between these 2 species and also another one that we had just morphology. So I felt more comfortable to actually pool all the data of these 2 genus to construct pooled P matrices that represent the within species patterns. So these are much well estimated matrices. So they were very similar. So we also have the between species morphology and performance data. So we have much more data for just morphology, as you know, it's can be called a curse too, Chris. So we have 61 species for morphology, but for 18 species across these two genera, we have also performance data, which is peak velocity and peak acceleration for jumping. And what we did, first we constructed divergence matrices for each of these genuses, assuming genera, assuming Brown in motion. So we can then determine did the evolutionary divergence followed these axes of most genetic variance, right? This is my first question, and I will say yes. So these are the pooled P matrices in visual representation, and these are the divergence matrices. So you can see they are very, very, very similar, but the correlations got a little bit tighter along evolutionary time. So the changes in these traits are tighter, so the changes are more correlated than you would see within species, but you see a very high alignment between Pmax and Dmax, axis of most divergence and most variation, and also for P2, so the second axis of divergence and the second axis of variation. So, okay, we found that persistent effect, actually, of trait correlations on divergence in this system, as had been showed for other systems. So now, I want to tell you what are these things. So I'm going to focus a lot on Pmax because it just explains so much variation within and across species even more. Is over 90% of the divergence of these species is on allometric size. So here's a presentation of where the species fall for both genera. So bigger frogs, smaller frogs. And the second axis, as you might expect, we can call it high limb shape. But it explains very much low variance and divergence. And this allometric size, what does it mean? It means that both individuals, because you see this within species too, so both individuals and species that are bigger have more muscle cross-sectional areas than individuals and species that are smaller. And this is kind of important for performance. But now I want to know, so I want to test my hypothesis. This axis of most variance aligns with selection line of least resistance first, and then with function, which I'm going to show you how I actually calculated this. So I need a model to calculate these selective lines of least resistance. So I'm not going to show the results here, but I rejected the drift hypothesis. So divergence on morphology in this system is not driven by drift. So now I assume a selection model in which the correlated changes in traits can be due to genetic correlations or co-selection of traits. And this is where I can get to my selection lines of least resistance. So I can reorder this equation, so discount the genetic correlations, and actually calculate the selective covariance matrix, this omega matrix, which gives me what are the traits that will be co-selected over time by directional selection, just in this case. And the eigenvectors of the omega matrix are the selective lines of least resistance. They are the major axes of the adaptive landscape, which I expect that could be aligned with Pmax in this system. However, no. So these are the pooled matrices, P matrices. This is a simplified leg length and total muscle conceptual area of the hind limb for both genuses. So you can see the upper diagonals are correlations. Positive correlations for these two traits in the phenotypic matrices, but in the selective covariance matrix, or it's negative, or is no correlation at all. So they are not aligned. They are not aligned. I am already rejecting my hypothesis of alignment between genetic variances and the adaptive landscape. So the second part, it was to see if the selective lines with resistance and Pmax will align with what I'm calling function. In this case, it's jumping performance. So I estimated performance gradients on the species that we could do that. Within and between, I'm just showing the results for between species because they were identical between and across species. And they reflect the form-function relationships, which is the symposium is about. So then I do vector correlations, and these are for the 6 hind limb traits, between the performance gradients and the major axes of the adaptive landscape, and also with Pmax, and they're totally unaligned. So the selection lines of least resistance and trait correlations do not reflect form-function relationships as I expected and wanted to test. So what do we do now? So we went further along and decided to directly model the evolution of jumping performances on the species that we could do this. So these are 18 of the species. And we ran models of trait evolution like Brownian motion, some adaptive models that you have 1 or 2 peaks. And what we found most support was for Brownian motion models, which we could interpret as not necessarily drift, because I rejected that already, but a fluctuating selection on jumping performance. But for maximal acceleration, we found an interesting pattern, which is this evolution being channeling mostly to allometric size had a consequence for jumping performance. So it basically seems that performance tracked the evolution of size. And an interesting pattern, which is that smaller species have higher maximal accelerations than bigger species, and this is probably related to some elastic energy that they're using more frequently than bigger species. So this channeling of divergence along Pmax actually had a consequence for the evolution of jumping performances across species, which I think is an interesting thing. So to conclude, this high alignment that we found between trait correlation and divergence was not explained by the evolution along selected language resistance. We found stability of trait correlations at a macroevolutionary scale. And because a lot of this Pmax and Dmax was on allometric size, we are suggesting that it's due to conserved developmental regulation of growth patterns. So this is what's determining the stability of the P or G matrix. This lack of alignment between Pmax, like trait correlations, selective language resistance, and performance might indicate that jumping performance is not a major contributor to fitness in frogs, which can be very weird. But it actually agrees with Daniel's previous work that across a huge range of species of frogs, they all are in the same peak of jumping. So they really jump very well, all of them. So, this could explain this. But actually, I rather think about this as support for a selection plus constraints hypothesis. So not a per selection, which means that you have fluctuating selection on jumping performance plus stability of trait correlations. And this is determining this divergence mostly along these axes of phenotypic variance. So this would agree with a very recent study that was trying to connect micro and macroevolution, which is Hostad et al. And also Ieda and McLaughlin had a very nice perspective on this paper. So I'm using their visual representation of this to explain our own data. So what does this mean is that we have this ancestral G matrix on this landscape that actually has an area of very high fitness, that you can move around here without losing fitness. But it's not the major axis of the adaptive landscape that is aligning this matrix. It's actually, you have the performance peak jumping around in this area, so it's not affecting fitness. But the population and species are trying to track this fluctuating selection. And it's mostly going to track it along lines of genetic variance if it's changing all the time. So you cannot actually get to the peak because it changed again, it changed again, it changed again. And so you're pursuing it along where you have most variance. So I think it kind of can fit well what we found if we consider that actually it's performance that is being fluctuating here in this area of high fitness, right? This means that the performance and the fitness landscape were actually not matched, but I think that's the case lots of times. So with this, I would like to acknowledge people that helped collecting the data on the field, museum curators, because a lot of this was morphological data, and Craig McGowan that helped us analyzing this performance data, and the funding agencies, of course. And if I have time, I don't know, I'm happy to take questions. Thank you. I probably don't have time.
I guess it's lunchtime, so
yeah, we can talk if you want. Or you can ask now, I'm available. Thank you. Do we need to do something here? No, right?
I don't think
I'm just going to close here this thing. I can't close it. Okay. Liked it? Fala.
I mean, I'm super interested in the whole, the macro bridge. So I work on alpha. I've seen a lot of different some different products. And I recently estimated gene editing in all of these species. So I'm sort of trying to look at integrating all of this macroevolutionary data in a macroevolutionary Thanks a lot.
Parce que si tu n'as pas de sélection, toutes les mutations sont là. Si tu as un processus de sélection qui se fait, juste par chance, tu vas juste perdre des lignées, mais aussi par sélection.